All right, next we have Olivia Asher from the University of Georgia. Her field is bioinformatics, and our advisor is Jeffrey Benetton. She did 2 practicums, both at Los Alamos National Laboratory. Let's welcome Olivia.
I'm super excited to talk to you today about fungi. So when most people think of fungi, they often think of something like mold, like maybe they left their bread out too long. They might think of mushrooms, either mushrooms they like to eat or poisonous mushrooms like this Amanita, or they might think of generally something that's decaying something. You know, it's breaking down a log or, you know, eating your bread before you can. But the fungi that I study do none of these things. These fungi they aren't a mold, they can't form mushrooms, and they also don't decay anything. These are arbuscular mycorrhizal fungi, these tiny microscopic fungi that live inside of plant roots, and then from their sort of home inside of the plant roots, they reach out into the soil and gather nutrients, like nitrogen and phosphorus. They get all of their energy in the form of carbon from their plant host. And a lot of times these fungi benefit plant growth by providing the plants with some extra nutrients that they gathered from the soil. But sometimes they can act as parasites if they take more carbon than nutrients that they give back. So these fungi grow in 90% of land plants, including a lot of plants that are important for crops. So my lab specifically focuses on bioenergy sorghum, which is a cereal crop that has potential to be used for biofuel if we can grow it large enough, cheaply enough. So that's where AMF comes in. Maybe we can use these fungi to promote plant growth without adding as much fertilizers. Now, the reason that people are not doing this is because the interactions between these fungi and their plant hosts are very complicated. And when I say AMF, these fungi, that's kind of like saying animals. So there's a lot of different types of animals with a lot of different functions, and that's kind of the same thing with these fungi. So just because there's AMF doesn't mean they're necessarily the most beneficial kind. Now, in addition to associating with plants, AMF also associate with bacteria. So here I'm showing a diagram of a cross-section of a plant root. You can see the orange fungal hyphae sort of snaking out into the soil. And on the surface of the soil, there are bacteria that also have properties that might influence the plant host, like fixing nitrogen, secreting phosphatases, which will make phosphorus more available. They might secrete plant hormones, and suppress pathogens as well. So now we kind of have this 3-way system with bacteria that we need to understand, the fungi that are really diverse that we need to understand, and all of these different crops. AMF also have bacteria that live inside of them. So these are 2 main types of bacteria known to live inside of AMF, Mollicutes-related endobacteria and Burkholderia-related endobacteria. It's not really known how they influence the fungus, but they are found in most of them. Actually, just as a side note, during one of my practicums at Los Alamos, I got to work with like a world expert on Mollicutes-related endobacteria, which is like kind of, I didn't know there would be a world expert on that, but there is. So endobacteria, external bacteria, is all seeming very complicated. But I have to be honest with you guys, I like fungi a lot more than bacteria. Sorry. So I forgot about this slide, never mind. Anyway, there's bacteria on the outside of the spores. Here's some pictures that I've taken of spores from the bioenergy sorghum fields in Georgia that show sort of like a rod-shaped punitive bacteria on the surface. So they're associated with the spores too. All right, now enough with the bacteria. Let's go on to the fungi. So I wanted to know, all right, so we have AMF, they're associated with all these different things. Are they also associating with other fungi that aren't AMF, like some of those molds or agents of decay that we talked about earlier? So I found some research on this from the early 2000s, where people did indeed find some fungi associated with AMF spores, but then they never really followed up on that ever again. So I was like, that's been kind of a long time. We have like more technology now. Maybe it's time to like re-look at these other fungi that associate with AMF, maybe they're really important to this symbiosis too, who knows? So I went ahead and investigated that. The way I did that was I got AMF spores from pot cultures, which are basically just a pot of plants that has AMF growing in it, and then you can isolate a lot of spores from there. And my pot cultures were made of soil derived from bioenergy sorghum fields in Arizona and Georgia. Then I isolated the spores, and you can see them here, all the little sort of orbs in the picture. And then I did DNA extractions on about 1,300 individual spores. And the way I do that is I isolate a single spore, I put it into a tube, and then I physically pop the spore and then heat it. And because I'm dealing with such like a small amount of input DNA in these experiments, I use a lot of negative controls to make sure that I wasn't getting contaminants instead of like things that were really associated with the spores. And then once I got that DNA, I subjected it to 16S and ITS amplicon sequencing. So that's basically just sequencing a very short region of DNA just to sort of get identities of different organisms that might be there. 16S will do that for bacteria, and ITS will do that for fungi. And I just wanted to show you a picture. So normally I would do this DNA extractions under a clean hood, but I generally don't take pictures then because I'm trying to be clean. But this is just a general look of what my setup would look like when I'm doing all of those DNA extractions. Got my microscope, everything going on. Then, once I get the data back, you get basically a huge amount of sequencing data, and this is going to require high-performance computing to analyze. So this is kind of my general workflow for anyone who's in bioinformatics and is very interested in this. But yes, the main important thing that everyone can take away here is that basically I'm taking the data, cleaning it, and then I'm identifying the sequences that I've got in my data set on a cluster. And I did this all using Sapelo 2, which is the University of Georgia's computing cluster. That's interesting because it's one of the few university computing clusters specifically designed for biological problems, which tend to be massively parallel. So it was nice to work on this computer. Now, as for some of my results, so remember I said I did DNA extractions on over 1,000 spores, but there's a very high failure rate with that, so I only got sequencing back from 281 individual spores. And in these spores, I found 5 different types of AMF. Only 45 of these 281 spores had other fungi present. And these other fungi that were present were highly variable across the individual spores. I kind of thought, oh, maybe I'll find like a fungus that's with a lot of the spores and it has some sort of interesting function, but that's really not what I found. I found something different on every single spore. And the other interesting thing was most of these fungi have never previously been seen associated with a spore. So to me, as someone who likes fungi, I was like, that's exciting. So here I'm just showing some basic cladograms to give you a little bit of the idea of the diversity of samples found. So Glomeromycotina is the name for the AMF, so you can kind of see how they are spread across the tree. And you can see that I found majority of spores by a vast amount from Enterophospora. This was not ideal news to me because I wanted to compare the microbiomes of different types of AMF, and that's not happening clearly because I have 281 in one group and 5 in the other group, so a little bit disappointing to me. And then over here you can see the broader fungal tree showing that we found other fungi associated with AMF spores from Ascomycota, Basidiomycota, and Chytridomycota. And Basidiomycota are like mushroom-forming fungi, so this is interesting. So we're finding like a broad variety of different fungi associated with these spores. Here is where some of these fungi we found have previously been found. So this was interesting to me because a lot of things on the bottom half of here are things that I expected to see. You know, a general saprophyte, plant pathogenic fungi, or fungi that just sort of generally associate with the soil. One thing that I didn't expect was to find a bunch of fungi that have only previously been found associated with rocks or cave walls. So I was like, well, that's interesting. I wonder, now we have some more information on the lifestyles of those type of fungi. And then remember, I had a huge amount of sequences from one genus, Enterophospora, so I wanted to look at the genetic diversity of those samples from the two sites that I looked at, Arizona and Georgia, which I had 5 different pot cultures. What you're looking at here is sort of an extremely sparse heat map where on whatever side that is, where it says cladogram, I have basically, that's showing the genetic relatedness of all of the different sequences that I got, and then in the heat map it's sort of clustering by the relative abundance. And what you can see from this is that there are some distinct sort of band groups of different types of Enterophospora that we found. What's interesting about this is it was not clustered by location or by pot culture. So no matter, you could find the same sort of type of AMF in Georgia or Arizona, which indicates there might be some sort of like long distance dispersal of these spores, which is something that's like very recently started to be investigated. And I strongly believe that this was not due to us, because the pot cultures from the Georgia soil and the pot cultures from the Arizona soil were developed in completely different states. So it's likely that this is like a real phenomenon we're seeing. So to move forward, I'm also going to be looking at the bacterial data, even though it's not my favorite compared to the fungi. I'm also going to be doing some full shotgun metagenomic sequencing of these spores to get a little bit more in depth on what's found there. And I'll do analysis on that and also plan to compare the genomes of the different genera of AMF that I found. And all of this research is very basic exploratory research, just sort of like looking at what's there. But in the end, we hope that this will contribute to efforts in sustainable agriculture when we want to look at specific systems and say what microbes are present in this environment, what do they do, and how can we sort of optimize this for optimal plant growth. We need to have this kind of information in order to be able to do that. So even though this really kind of gets down a rabbit hole of what fungi are on an individual AMF spore, I do think that basic research is really important to lead to applications later on down the line. So all of this work was sponsored by the Department of Energy, including CSGF, and another DOE award that was given to my advisor. So thanks so much.